Hello, and thanks for joining us today for this Arima Genomics webinar on how HiChIP is changing cancer research. My name is Christian Nelson, and I'm Arima's Global Sales and Support Director, and I'll be the moderator for today's webinar. Thank you in advance for your time and attendance. So let's get started. Our first speaker today is Anthony Schmidt from Arima Genomics. Anthony joined us here at Arima in 2016 and serves as the Senior Vice President of Science. He has led the development of the core Arima-HiCIP chemistry and technology platform since Arima's inception, contributed to core inventions and technology development grants, and continues to lead our R&D programs for targeted chromosome conformation capture techniques such as HiChIP and Capture-HiC. Prior to Arima, Anthony received a PhD in biomedical sciences from UCSD in the laboratory of Dr. Bing Ren, and a bachelor's of science in biology from UMass Amherst. During his doctoral training, Anthony played roles in developing and applying HiCIP technologies with notable contributions to our understanding of gene regulation in nearly 30 human tissues using HiC and promoter Capture-HiC, developing target haplotyping methods for medically relevant loci using Capture-HiC and HaploSeq, and finally developing PlaqueSeq, also known as HiChIP, for discovering long-range gene regulatory interactions associated with specific chromatin proteins. Anthony, we appreciate your time. Good to see you. The floor is yours.
All right, wonderful. Thanks a lot, Christian. Welcome, everybody. A warm welcome from us here at Arima. My name is Anthony Schmidt. Today, I'm just going to spend about 15 minutes providing an introduction to Arima, a little bit of background about 3D genomics, and kind of go through one example of why you might want to start thinking about using those approaches if you haven't already. And then I'll spend a bit of more time diving deeper into HiChIP specifically and some of the nuts and bolts of that technology to sort of set the technology landscape for the wonderful talk that you'll hear from Lindsay after myself. I am going to shut off my video just to preserve some of the bandwidth on my home Wi-Fi, but just wanted to say hi to everybody and put a face to the name. So a little bit about Arima Genomics for those who are new to Arima. We are a biotech company that was spun out of Dr. Bing Ren's lab back in 2015 and 16. And our real mission at Arima is to accelerate the understanding of the 3D genome and how this 3D genome folding information plays a role in uncovering fundamental discoveries in human health and disease, as well as species evolution. And we try to do this by really democratizing access to what we think is this powerful 3D genome technology for basic and translational research applications through reagent kits like the one you see here on this picture, as well as services. We think we are on our way towards reaching that mission with over 70 publications and preprints now using our technologies. So back in 2018, just a little bit about Arima's technology platform. We launched our first HiC kit, which you can see here, which is a tool for genome-wide assessment of the 3D genome and chromatin conformation. And of course, genome-wide assays are not the most appropriate for every single biological question and to test various hypotheses. So we have since made available 2 different targeted versions of chromatin conformation capture. The one on the right here is called Capture-HiC, and that's enriching for various sequence elements using capture probes and then measuring the 3D interactions associated with those enriched sequence elements. And then here on the left is what we'll be talking about more today, which is Arima-HiChIP, which is also a targeted version of HiC, except you are enriching using an antibody, and then what you're uncovering really are those chromatin interactions that were associated with the immunoprecipitated protein. And just as a brief outline, I'll say a few words about the 3D genome introduction and some motivation behind why that's important. And then I'll talk more about Arima-HiChIP specifically and some of the nuts and bolts about the workflow and the informatics, and maybe 1 or 2 data slides. So 3D genome introduction, as Christian said, this is my own personal background, and these tools have been used for a long time. Specifically, the chromatin conformation capture sequencing-based approaches have been used for the past 15 years and have made a pretty broad impact across many different research areas, Some of those examples are just shown here from Effie Apostolou's lab, Arthur Golchi's lab, Roger Fu's lab, the Phillips Crimmins lab at Penn, the Akbarian lab at Mount Sinai, and then the Sirigos and Iofantis labs at NYU. So it's really made a broad impact across many different research areas, and I just wanted to tell a quick story about some of the impact that it's made in cancer research leading up to what we'll hear more about from Lindsay and her story. So just to take a deeper dive into this study from the Iofantis and the Sirigos labs, what they did in this study is they generated Hi-C, genome-wide Hi-C data sets on a cohort of T cells isolated from healthy human subjects. And what you're looking at here is basically a Hi-C interaction matrix that's zoomed in around the MYC locus. So here's the MYC gene. The diagonal here is the linear genome, and we're looking at about a 3 megabase window around the MYC gene. And the two things that I wanted to point out that you can see from this type of Hi-C map is that the MYC gene lives right here within this self-interacting chromatin domain, or TAD, as they're often called. And then in a neighboring self-interacting domain, hopefully you can see my cursor, are some known super enhancer elements that are in a separate domain. So you could infer from the Hi-C map that they're sort of spatially segregated from the MYC gene, right? And for those of you who are new to looking at these types of Hi-C maps, schematically what we're talking about here is in healthy individuals, you have your MYC gene here in orange and your super enhancer element here in blue, and they're in two neighboring but spatially distinct chromatin domains. Now, in the analysis of primary T-cell acute lymphoblastic leukemia cells, as well as a leukemia cell line, when you do Hi-C analysis and you look at the same exact region, the striking thing that really pops out from this image is really the coalescence or the merging or the fusion, as they say in the paper, of these two neighboring topological domains into one sort of larger, broader self-interacting domain. So you might think that that could provide the sort of spatial flexibility for the enhancer elements if they were turned on to then interact with MYC and drive oncogenic MYC expression, right? So from the Hi-C data, you can see that these two regions are now interacting more. And then again, for those who are not used to looking at these Hi-C maps, this is kind of what we're talking about schematically here between the MYC gene and the enhancer element. Right, so this is the Hi-C data. Now, if you look at the underlying chromatin and transcriptome features, what does that look like? Well, in the healthy T cells, shown here in purple, if you look at CTCF binding, for example, what you see here is that you have CTCF binding really on the far left and the far right of this region. So in healthy T cells, that would be here. And in healthy T cells, you also have CTCF binding right here in the center, which makes sense because CTCF is a protein that's known to demarcate the boundaries between those topological domains. And what you also see when you start looking at the leukemia lines is that really the lack of binding of CTCF right here in the center, which also sort of corroborates what you see in the Hi-C data with the coalescence of those two neighboring domains. Similarly, when you look at the K27 acetylation signal for the enhancer marks in the healthy T cells, really no enhancer signal downstream of MYC. So MYC is oriented right here and about 2 megabases away from that or so would be the super enhancer regions. And then in the leukemia cell lines, now you have the enhancer signals come on. And lastly, when you look at the MYC expression levels, it's also more in the leukemia cell lines. So really taking this together, the Hi-C, the chromatin, and the expression data gives you this insight into how the 3D DNA folding information, right, regulates MYC expression in a healthy state, keeping the expression relatively low, and also the 3D DNA misfolding information that you obtain from Hi-C analysis on the leukemia lines tells you something about how MYC is misregulated in the leukemia cells. So with that in mind and sort of a motivation for us at Arima of why we develop these sorts of tools and the discoveries that that they can enable, I want to switch the focus really to talking about not just 3D genomics in general, but specifically the HiChIP technology. All right. So in the second half of this introduction here, I want to start with really just super high level. Where does generally HiChIP fit into the overall sequencing sort of workflow? And really HiChIP comes up here as a sample preparation technique, much like ChIP-seq. Except with HiChIP, what I'm going to tell you in a second is that you capture the 3D genome information and really the chip information in this upfront sample prep stage. Then you go through your Illumina library prep, your Illumina sequencing, and then at the end, Arima-HiChIP bioinformatics, which again is designed to extract both the chip information while simultaneously extracting the 3D genome information in a single pipeline that I'm going to tell you all about. Okay, that's high level. Now, kind of peeling back the layers here, I want to just give kind of a molecular workflow for those who are not familiar with HiChIP. I like to introduce it really first and kind of position next to ChIP-seq, which is a bit more ubiquitous. So in ChIP-seq, as many of you know, you start with cross-linked chromatin here, and then with a few steps, you just shear that chromatin into smaller pieces, and you add your antibody, and you do your immunoprecipitation reaction. And that immunoprecipitated complexes, you add your sequencing adapters to those molecules that are enriched, amplify, and then sequence, right? Now, HiChIP is really almost exactly like it sounds. It's really the sequential marriage of Hi-C and ChIP-seq. And I'll step through this somewhat slowly. You start with the fixed cells, just like you have for ChIP-seq. And then there's really just 3 core steps that you do to capture the 3D genome information. And this is really the core Hi-C technology that we've built at Arima, are these core 3 steps. We digest with the restriction enzyme cocktail. We label those ends with a biotinylated nucleotide, and then we ligate them back together. So really up to this point, you have 2 pieces of DNA that were held together in 3-dimensional space. And now they've actually been physically adjoined. And that's sort of how you capture the information that they were spatially proximal to one another. Okay, so you carry out Hi-C, and then when you have this proximally ligated chromatin, you shear that and you add your antibody. So at the molecular level, when you compare what's being enriched from ChIP-seq up here at the top to HiChIP down at the bottom, it's still the same protein binding sites, right, that you would get from ChIP-seq. The difference is that those DNA molecules that are bound to that protein also contain the 3-dimensional information that was captured during the upfront Hi-C, right? So when you just look at this at the molecular biology level, you can see how the HiChIP assay is detecting those interactions that were associated with the immunoprecipitated protein. Okay, so you do your IP reaction, as I said, you enrich for the ligation junctions through a simple biotin streptavidin conjugation here to capture those interactions. You add your sequencing adapters, you amplify, and you sequence. And again, these final molecules that get sequenced ultimately had to come through 2 enrichment steps. They had to be enriched with the antibody, and they had to be proximally ligated events that were captured during the upfront Hi-C step. So in sum, this technology is really designed to inherently measure the 1D protein localization sites that you would get from ChIP-seq, and that Lindsay is going to also talk about, and the 3-dimensional interactions with those immunoprecipitated proteins. So think H3K27 acetyl, H3K4 trimethylation, and characterizing those interactions that were associated with those active chromatin marks. We have optimized chromatin sharing protocols, and we offer this as a kit or service format. So diving into some of the nuts and bolts of the bioinformatics, first I just want to say that we've had a wonderful collaboration with Dr. Ming Hu at the Cleveland Clinic over the past 1.5 years or so of our HiChIP technology development. All of our HiChIP analysis tools are open source. Down here is a GitHub link to find the location of this pipeline. And really this pipeline was originally developed by Ming Hu, and through our collaboration, we've built some additional capabilities on top of that pipeline. And really, you start with your Arima-HiChIP FASTQ files. They get fed into an alignment program called Feather. So that maps the HiChIP data, and the output of those alignments are used really to call the chip peaks from the HiChIP data. And then you bring together the chip peaks and the long-range interactions from the HiChIP data, and you call statistically significant chromatin loops using a statistical framework called MAPS. So that's how you derive your significant interactions. And then as an output of this pipeline, you get a nice quality control table. You get a chip enrichment meta plot to really make sure that your data is enriched for the factor that you enriched for. And then lastly, you get these really nice arc plots that you can upload to the WashU genome browser. They are color-coded. The darker the arc, the more significant the FDR value of a particular interaction. And then you can view these alongside your other omics data sets. Here's just the one data slide that I will go through. This is also as an example of the MIC locus from the WashU genome browser. There's lots of different chip tracks here, CTCF ChIP-seq, cohesin ChIP-seq, et cetera, RNA-seq. And then down here on the bottom is what I want to draw your attention to. This is 2 replicates of H3K27 acetylation ChIP-seq data from ENCODE. And if I show you now the H3K27 acetylation HiChIP data plotted as this sort of one-dimensional signal track, it looks like this. So it very much replicates the ChIP-seq data in terms of the signal to noise, the peak positions, and the peak heights. We've done quite a bit of chip peak calling analysis that I can talk about more in the Q&A session if that's of interest. Right. So you get that binding profile in HiChIP that very much recapitulates ChIP-seq. But then, of course, what you're not getting from the ChIP-seq data alone is all the 3-dimensional connectivity information between these active K27 acetylation peaks. Right. So here's an example where you have MIC right here, which is known to have several interactions upstream of MIC to these other K27 acetylation sites in human lymphoblast cells. This is the type of information that you can get. And I should say we've been thoughtful about reproducibility between these loops that are called in 2 different replicates. They look, qualitatively similar here. And when you quantify that at a genome-wide scale, you in fact see that there's about 85% reproducibility between replicates in our K27 acetyl, and a similar story for H3K4 trimethylation. So in sum, HiChIP is a multi-omic approach for chromatin protein binding profiling as well as chromatin interactions. I've spoken about the reproducibility. I didn't have time to show data from tissues or blood or other factors like CTCF, but I can answer that in the Q&A. Spoke about our open source bioinformatics pipeline, the customer validation across many different factors and cell types, and we have expert assay and bioinformatics tech support to help you along the way. And with that, I'll hand it back to Christian. Thank you very much.
Perfect. Thanks, Anthony. Really appreciate the background. I know you have quite a few tricks up your sleeve for product launches this year as soon as June, so looking forward to that as well. Now on to it. Our featured speaker today is Dr. Lindsay Montefiore from St. Jude Children's Research Hospital. Dr. Montefiore received her PhD from the University of Chicago in the Nobrega lab of the Department of Human Genetics, where she used promoter capture Hi-C and other Arima technologies to map cardiovascular disease GWAS loci to target genes. She is currently a postdoctoral fellow in the Charles Mulligan lab at St. Jude Children's Research Hospital, Department of Pathology. Dr. Montefiore is focused on modeling genomic alterations in lineage ambiguous leukemia. Thank you for your time, Lindsay. We really appreciate it. The floor is yours.
Great. Thank you for the invitation to speak at this Arima seminar. So as Christian said, I'm Lindsay. I'm a postdoc at St. Jude and Charles Mulligan's lab, and I'm going to share a story from our lab that's focused on identifying this new subtype of lineage ambiguous leukemia, where we've been able to use HiChIP data to get at the genomic mechanism underlying the formation of this distinct type of leukemia. So acute leukemia is the most common childhood cancer. And this slide is just to show that, as you probably know, leukemias arise from hematopoietic cells, so blood cells. And most leukemias are pretty standard to diagnose. So they're diagnosed based on their immunophenotypic similarity to normal hematopoietic cells. And that is shown here, where B-ALL or B-cell acute lymphoblastic leukemia is identified because it expresses markers that are found on normal B cells, for example, CD19. Whereas T-ALLs generally express CD3, which is found on all T lymphocytes. And the other main lineage is myeloid lineage. This is your innate immune system. And acute myeloid leukemia typically expresses markers such as MPO, which is myeloperoxidase. This is the enzyme that's produced by innate cells to clear up infections. It's kind of what's responsible for turning your pus yellow when you have an infection. But it's a really handy tool for identifying myeloid lineage leukemias. However, there's a subgroup of leukemias, which is the focus of this talk, that are collectively known as acute leukemias of ambiguous lineage, or ALAL. And these are thought to arise in immature, uncommitted stem and progenitor cells at the top of this hierarchy. This includes acute undifferentiated leukemia, which by definition do not express any lineage-defining markers. Most frequently, it occurs as mixed phenotype acute leukemia, which express markers associated with both the myeloid and the lymphoid lineage. And furthermore, early T cell precursor acute leukemia is recognized as a subtype of T-ALL, but these leukemias look very similar to stem cell and mixed phenotype acute leukemias. Really, the only distinguishing feature is that they don't express MPO. And so this is just an example of what one of these leukemias look like. This is a flow cytometry analysis of a primary T myeloid impal patient sample. So on the x-axis is staining for myeloperoxidase, which is the myeloid marker. And on the y-axis is CD3 for the T cell marker. And you can see that this single leukemia sample has populations that express both of these markers or only one marker. And so we were motivated by this to identify biological heterogeneity among acute leukemias of ambiguous lineage, because like I said, sometimes they express myeloid markers or T cell markers or both. But then also we want to understand what are the molecular drivers of this plasticity? Why do we see these different populations within leukemias? And this really hasn't been worked out yet. So the approach that we took, I'll motivate that here. This is work from our lab published a couple of years ago on B-ALL, which was thought to just not really be that heterogeneous. But as you can see, based on transcriptional profiling, there are actually more than 20 distinct subtypes or subgroups of B-ALL. So these different clusters you see here, where each dot is a patient sample. They're color-coded based on genomic alterations that have been identified, and these clusters correspond to these genomic alterations. And so this, I think, really nicely illustrates that there is tremendous heterogeneity among single diagnostic entities, and that the root of this lies in different sort of founding genomic alterations. So we took this approach to better characterize the lineage ambiguous leukemias. So for this, we compiled more than 1,000 transcriptomes from non-B leukemias, essentially. So T-ALLs, acute myeloid leukemias, MPAL, and ETP-ALL. And you can see here that the samples are clustered according to their original diagnosis. And in the kind of magenta and red color are MPAL and ETP-ALL, and they kind of follow all over the place. But what stood out to us is that there's this really distinct gene expression group circled here that's comprised of at least 4 different diagnostic entities. So AML, T myeloid MPAL, ETP-ALL, and then a group of samples that does not have enough information to know whether it's an ETP-ALL or not. But all of these cases exhibited monoallelic expression of a gene called BCL11B. This is a T cell transcription factor gene that I'll get back to in a second. And furthermore, we have whole genome sequencing data for the majority of these cases. And in every single one of those cases, we identified structural variants that are targeting the BCL11B locus. And so that suggests that could be sort of the founding alteration for this group. And just to reiterate, for those who are more clinically minded, this single biological entity, if you will, is comprised of multiple different diagnostic entities. So I'll spend the rest of the talk discussing these structural variants that we identified. So here I'm showing the B11B locus on chromosome 14, and each black tick that you see is a breakpoint for a different patient sample, and they are grouped based on the rearrangement partner. So the majority of patients harbored rearrangements to a region on chromosome 6 near the ARID1B gene, and those are highlighted here. The next most frequent alteration was not a translocation. This was a focal amplification of a short, around 2 kilobases DNA element that's located around 700 kilobases downstream of BCL11B, and I'll come back to that later. But I just want to point out that all of these breakpoints, as you can see in this zoomed in plot at the bottom, they, for the most part, spare the BCL11B gene itself. So these are non-coding breakpoints that occur either up or downstream of the gene. Okay, so why is this interesting? I mentioned that BCL11B is a transcription factor that is critical for T cell differentiation. And if you look at where this gene is normally expressed in hematopoiesis, this is shown here. This is a plot of all main hematopoietic cell types. And the cell types are colored and sized based on the level of BCL11B expression. And you can see it's more or less restricted to T cells. However, the hematopoietic stem cells and progenitor cells, which are circled here, are the hypothesized cell of origin for this group of leukemias. But BCL11B is normally repressed in this cell type. And so this led us to hypothesize that the structural variants that we've observed serve to ectopically or aberrantly activate BCL11B through enhancer hijacking. So we investigated this initially by checking the chromatin landscape of the rearrangement partners to see whether there was evidence of enhancers being present there in normal progenitor cells. And so here I'm showing you the ARID1B locus on chromosome 6. This is the most common rearrangement that we observe in this group. And highlighted in yellow are all of the breakpoints for patient samples with this rearrangement. And they cluster near this very strong super enhancer that's located quite far from the ARID1B promoter, but it's very distinct. And I'm not showing these data, but this enhancer in particular is specific to the normal CD34 positive hematopoietic stem and progenitor population. And so sure enough, when we looked at the remainder of the rearrangement loci across this cohort, every single rearrangement group harbored at least one super enhancer in normal hematopoietic stem and progenitor cells. So super enhancers, as I'm sure everyone knows, are just the strongest and broadest domains of H3K27 acetylation in any genome. And so you can see the quantification of this on the right. And so this is just to say that this provides evidence that these rearrangements are most likely resulting in enhancer hijacking. But of course, we want to demonstrate that with more evidence. And so this is when we turn to the HiChIP method that Anthony just described to you. So I won't go into details, but we use the Arima-HiChIP kit from Arima to perform H3K27 acetylation HiChIP in multiple patient samples. And so I'll just start by walking through what the data look like to give you a sense so that when I compare to the patient samples, it is more visually appealing. So what you're looking at here is the region of chromosome 14 containing the BCL11B gene. Its promoter is highlighted in yellow. And the blue track that you see is regular ChIP-seq for H3K27 acetylation. And these data are generated all the time, and you'll find peaks that are kind of in the middle of nowhere, and you don't know which gene this enhancer is regulating. This enhancer in particular has been elucidated. It produces an oncoding RNA called ThymoD that has been shown by others to be critical for normal BCL11B activation at the earliest stages of T cell differentiation. And there's a subtype of TALL actually that harbor rearrangements that result in this enhancer driving overexpression of the TLX3 oncogene. But those rearrangements weren't identified in this group. And so I'm just simply showing this here to illustrate that when we add in HiChIP data, we can very clearly see that this enhancer is indeed regulating the BCL11B gene. As you can see, this black arrow is pointing to a very intense interaction at this intense signal off of the diagonal, indicating that there's physical contacts between the chromatin domains consisting of the enhancer and the B11B promoter. And these two elements are 1 megabase apart. So this is quite a hefty long-range interaction. And I also want to reiterate something Anthony said, which was that the HiChIP data, the one-dimensional coverage track looks very similar to the ChIP-seq. So it's nice that when we do this experiment, we can not only capture the 3D conformation, but we can also capture just a normal H3K27 acetylation landscape data. And so I'll just contrast this to what this region looks like in normal CD34 hematopoietic stem and progenitor cells. These are undifferentiated hematopoietic cells that we isolated from cord blood. And you can see that there's basically nothing going on at this region. And that's because B11B is repressed in these cells. So now I want to show the chromosome 14 region next to the chromosome 6 region, because I'm going to, in the next slide, show you what this landscape looks like in a patient sample harboring this rearrangement. But first, just to contrast with the normal landscape. So this, again, is CD34 cells, normal stem and progenitor cells. The B11B gene is highlighted in purple, and there's no H3K27 acetylation, no long-range interactions. This is the ARID1B enhancer that I mentioned earlier, where most of the breakpoints occur near in proximity to this element. And you can see evidence that this enhancer does loop to ARID1B. So now if we look at a primary patient sample harboring this chromosome 6 14 rearrangement, you can see very strong evidence that there are several intense interactions between multiple enhancer peaks at the chromosome 6 locus that extend along the entire length of the B11B gene. And I just like to point out that this gene is 100 kb long. So this line that you see here means that this entire peak is interacting with the entire length of the gene here. And so these are identified as interchromosomal read pairs because we map to a reference genome. So these read pairs map to chromosome 14 and chromosome 6, but the patient-specific genome would have these insist with each other. And I also want to highlight just an interesting observation that if you just look at the interactions coming from chromosome 14, so these are just cis interactions along the B11B gene, we see this really strong H3K27 acetylation domain shown down here, and there's interactions spanning the entire length of the gene. And if we just contrast that with what this looks like in JERCAT T-ALL cell line, which also expresses B11B, but it's not necessarily oncogenic, you can see that looks very different, where there's only interactions between the 5' and 3' UTR. So I'll just point out that we've generated these data in 2 other patient samples, and in all 3 of these cases, we found evidence of enhancer hijacking. So in the primary patient sample, we could demonstrate that these progenitor cells, super enhancers, were also active, remained active in bona fide leukemia, and that they formed physical chromatin interactions with the active B11B gene. So remember, there's another allele of B11B, and it's repressed in all of these cell types. Okay, so I want to now talk about this interesting subset of cases. So around 20% of cases harbored a high copy tandem amplification that's located 700 kb away from B11B, and that is shown here. So this is zoomed in this region highlighted in orange. These are whole genome sequencing coverage tracks showing this sort of peak here. And so here are the break positions of the breakpoints across patient samples. And this wall of heightened read depth indicates that there's 15 to 20 copies of this element in these genomes. And at the bottom, you can compare what this looks like in a sample that lacks the amplification. So interestingly, we also noted that we can see RNA-seq signal emanating from this region, which actually allowed us to identify 2 additional cases for which we didn't have whole genome sequencing data available. And so this kind of gives a clue as to what this is. Clearly we think that this is an enhancer, and this transcription signal supports that it's enhancer RNA. So we wanted to resolve better the structure of this amplification. Just because we see many copies from whole genome sequencing doesn't necessarily mean that it's copied in tandem. So we performed long read sequencing using PacBio to try to resolve the entire length of this locus. And so I'm showing the results from 2 patient samples. At the top, you can see the whole genome sequencing coverage track. And then I have kind of a schematic for what this would look like if this element were copied, 15 times in a row. And the bottom shows a single read for each case. In one case, we actually didn't have any reads that span the entire length because it was almost 50 kb long. But you can see on the left in brown, there is a read that spans the entire length of this amplicon. And these dashed lines in a row just indicate multiple copies of this, what we're calling repeat unit. So, PacBio data has supported that this is a tandem array of this 2.5 kb element. And we're calling this the B7B enhancer tandem amplification, or beta for short. So in contrast to the other cases that I described previously, which we found very robust evidence that rearrangements occur in proximity to existing super enhancers, if you look at the H3K27 acetylation landscape of this element in normal progenitor cells, what the blue arrow is pointing to is, basically a very dismal K27 acetylation peak. And so we hypothesized that, normally this element is a weak enhancer, maybe. It is interesting that it's only present in progenitor cells still, even though it's really low. But following 15 times amplification, the signal would be expected to get a lot bigger, and that could generate sort of a de novo super enhancer from something that was otherwise basically, maybe a shadow enhancer or something. We don't really know. So we performed HiChIP in 2 cases with this beta amplification to determine if this region exhibited high levels of K27 acetylation and if it interacted with BCL11B. And so the coverage data for those experiments is shown below. You can see that the 2 cases shown here that have this amplification have remarkably high levels of H3K27 acetylation. So even when you account for the fact that this has been amplified 15 times, it's still a remarkable enrichment compared to other similar K27 acetylation peaks in the genome. And just to say that this is uniquely identified in these cases as compared to the other 3 primary patient samples that we examined, normal CD34 progenitor cells and 2 T-ALL cell lines. So this is really quite a unique event that might represent a new way of driving oncogenic expression in cancer. So then, of course, to see whether this region interacted with BCL11B, we can look at the 3-dimensional data. And so indeed, beta is looping quite strongly with, again, the entire length of the BCL11B gene. And we also find evidence that it loops to this enhancer that I mentioned earlier, this thymo-D element that is normally only active in bona fide committed T cells. And so we speculate that this, following its amplification, this beta element might be capable of also sort of waking up other dormant enhancers nearby to collaborate to drive strong BCL11B overexpression. And so just transitioning through to the data from that T-ALL cell line, you can see that this beta element is normally, as I said, not active. And then in a primary patient sample, in this case, this sample harbored a rearrangement to an enhancer at the distal end of MYC that I think Anthony touched on earlier. And you can see that even though these cells express BCL11B and this gene is active, as you can see in the bottom, these 2 elements are completely off. And the reason why this gene is active in this case is because there's another super enhancer that's come in from chromosome 8 that's driving this upregulation. Okay, so I think that I've shown the power of HiChIP to resolve these interesting structural variants that we've identified that sort of define this new subtype of lineage ambiguous leukemia. But we really wanted to know what could BCL11B be doing to drive this. This is a T cell transcription factor. It's not mutated. It's not a fusion gene. So how could ectopic BCL11B expression be oncogenic? And so just to initially investigate this, we are taking an overexpression approach in healthy human CD34 progenitor cells. So we have obtained cord blood from 3 different donors, where we can isolate the CD34 population, transduce them with lentiviral vectors encoding B11B or an empty vector, and then the data that I'll show are transcriptional profiling and the impact on in vitro differentiation. So here are the results of the RNA-seq analysis. So again, comparing the 3 donors overexpressing B11B to 3 donors that that had just an empty vector. This was only 4 days after transduction, so it's kind of an acute time window of looking at the impact of B11B on gene expression. And we actually found quite a lot of gene expression changes. So there are around 600 genes upregulated with a fold change cutoff of 2, and around 550 downregulated. And I just want to point out that, so shown here is the CD3 gene. So CD3 is that key diagnostic marker that helps distinguish T lineage leukemias. And it's the T cell receptor co-receptor that is composed of multiple subunits encoded by the CD3 DEG genes. And so here I'm showing you that CD3G is upregulated following B11B expression. And the other members of this receptor are also upregulated. I'm just not pointing to them. So gene set enrichment analysis is a way to globally look at what sort of biological pathways might be reflected in these gene expression changes. So on the left, I'm showing you that the upregulated genes are highly enriched for gene sets related to T cell differentiation. And on the right, I'm showing you that the downregulated genes are negatively enriched for genes related to myeloid differentiation. So basically, after putting in BCL11B, so ectopic B11B expression results in activation of T cell genes, repression of myeloid genes. And this might not be too surprising given that this is a T cell transcription factor, but it's just interesting to think about why we see specific immunophenotypes in different leukemias, and getting back to whether it reflects the cell of origin of the leukemia, or the cell is actually trying to differentiate, or it's just completely aberrant, and it's because there's a T cell transcription factor that's activating these immunophenotypically relevant genes. So we next tested differentiation of these cells in liquid culture, and so I'll just show one little snippet of this. So we have, in this case, transduced with lentiviral vectors encoding B11B together, so with or without FLT3 ITD. So I didn't mention this earlier. FLT3 is a kinase molecule that is commonly has activating mutations, so these internal tandem duplications in myeloid leukemias. And we found that this alteration occurs in more than 80% of this B11B rearranged subgroup. So we think that there's definitely some synergy between these 2 alterations. And so we wanted to model that here. And I want to point out that in the condition where you have both B11B and FLT3 overexpression, you get this population of cells that expresses cytoplasmic CD3. So this supports the RNA-seq data where we saw upregulation of CD3 genes, but it also supports that B11B together with FLT3 might be sufficient for driving this immunophenotype that we observe. And normally you only get these genes being activated because the cell has immigrated to the thymus and it receives inputs from Notch signals. So in this case, this was in a dish without any Notch signaling, and that was B11B was able to independently drive this immunophenotype. So finally, we wanted to assess transformation properties. And it's difficult to do this in normal human cells. So for this, we turned to the mouse system. So we isolated lineage negative hematopoietic stem and progenitor cells from normal mouse bone marrow, transduced them with those same constructs. So with or without B11B, with or without FLT3 ITD. And we did an assay called colony forming assay, where we plate cells in a dish with methocol, which is a semi-solid medium with various cytokines to promote myeloid growth. And then we count how many colonies we get. Then you harvest all of that and you replate them. And you do this multiple times. And if a gene or a mutation is oncogenic or transforming, you'll get serial replating. Whereas if it isn't, the cells will eventually just stop replating because they terminally differentiate. So this is a surrogate for self-renewal and transformation. And what we see very interestingly is that only in the condition with BCL11B plus FLT3-ITD do you get meaningful serial replating as shown in these blue bars here. And you even with BCL11B only, the gold bars, you do get some evidence of replating compared to the control or FLT3 only. And so this provides evidence that BCL11B, this ectopic BCL11B activity in a progenitor cell can be transforming. So I'll just bring it back to the beginning of my talk. The motivation for this was on the left to gain a better understanding of biological heterogeneity that exists among these lineage ambiguous leukemias, and then to identify molecular drivers of this lineage plasticity. So I'll just summarize the main points of this talk, which was that we were able to use gene expression profiling to identify a new subtype of lineage ambiguous leukemia. And these are defined by structural variants targeting the BCL11B gene. And importantly, this is comprised of multiple diagnostic entities, so including up to 40% of all leukemias currently diagnosed as T myeloid impal or ETP ALL, and suggesting that these diagnostic distinctions aren't always able to accurately reflect the biology of the leukemia. We used HiChIP in primary patient samples to demonstrate that these rearrangements result in enhancer hijacking of super enhancers that are normally active in hematopoietic progenitor cells. We identified a new oncogenic event, which is a high copy tandem amplification downstream of BCL11B that drives BCL11B overexpression. And this is actually quite common, occurring in 20% of these cases. And then finally, through modeling of ectopic BCL11B expression in human and mouse progenitor cells, we're able to demonstrate that BCL11B is sufficient for driving different gene expression profiles consistent with the T lineage immunophenotype that you see in these leukemias, and that there's evidence from the mouse data that BCL11B can be transforming. So I'd just like to acknowledge everybody who is involved in this work. This is quite a large scale project with many people and collaborators involved. So I just want to thank them all here, in particular my PI, Charles Mulligan, and Zhao Wei Gu, who did all of the gene expression profiling in the beginning. And thank you all for your attention.
Perfect. Thank you, Lindsay. We really appreciate your time. We do have some questions. Before we go, just wanted to remind everyone that we have just days left in the availability of our HiChIP grant program. Please find the grant application link on the homepage of arimagenomics.com. It's a great way to pilot this technology. Jumping into the Q&A, a couple of questions in a row here for Anthony. Let's see, why don't we start with this one? So is it possible to apply HiChIP to different model organisms, mice, chicken, zebrafish, et cetera? And would general or human antibodies still work?
Maybe there's a couple ways to answer that. Just from our own in-house experience, was primarily with mouse and human. So yeah, I can speak to that and say that HiChIP would work just fine with mice. In terms of other organisms, I know that we've had customers who are trying things that we haven't internally validated, like Drosophila or even plants, for that matter. We've tried to build the assay with experimental QC checkpoints to sense sort of whether the assay is working or not. And then we also recommend shallow sequencing, I guess, after you do your experiment. And the bioinformatics pipeline is designed in such a way that even with shallow sequencing, you should be able to look at the 1D signal enrichment, the percent of long-range interactions captured, et cetera, to kind of tell if you're on the right track before investing in deeper sequencing. And then I guess lastly, I think it would be fair to say that if you're working in a model organism where a human antibody works for ChIP-seq, then there's a good chance that that'll also work for HiChIP. But we would still recommend that you test it out empirically and make sure that it works first.
Okay, great. I think we might have touched on this earlier in the talk, but the question came up pretty early as well. Is it possible to identify super enhancers using HiChIP?
Yeah, we've never used the computational tools to specifically call super enhancers. Like I think there were some of the plots that Lindsay showed that are used to derive sort of the super enhancer computational definition. We've never done that. I don't know, Lindsay, if you actually tried to do that from the HiChIP data, but my sense would be that if the 1D binding profile between HiChIP and ChIP-seq is very similar, then you should be able to.
Yeah, we haven't, this has been something I've thought about doing and just kind of haven't done it yet, but I think it would be possible to identify them, because the 1D track does look so similar.
Excellent. And Lindsay, while I have you, maybe a couple of questions for you. Let's see, maybe the first could be, have you examined where BCL11B might be binding in these samples?
Yeah, so we've done ChIP-seq for B7B in a few of these patient samples, and we're still sort of analyzing the data, but the interesting thing that we have found is that B7B seems to be binding quite widely. So there are around 10 to 15,000 promoters with evidence of B7B binding in these leukemia samples. And then if you look at the distal regions where it's binding, there's enrichment for Runx motifs, which has been described in MPAL single cell ATAC-seq data before. So we're not quite sure what to make of that widespread binding. Kind of makes it difficult to get at what are the target genes. But yeah, hopefully we'll have more soon.
Okay, great. And then just thinking of translating this to the clinical setting, if this leukemia subtype is spread across different diseases, how will you be able to identify them in a clinical setting?
Right. So I think at the moment, the most straightforward way to identify these would be, I guess there's two methods. Either you do RNA-seq and you're able to perform that clustering analysis where you can see that it falls in that cluster, or you do, an whole genome sequencing to identify these structural variants, which is not really, applicable to a routine, I don't think, diagnostic setting. But that really strong overlap with FLT3 alterations might be the best way. So if you have a diagnosis of ETP, ALL, or a T-myeloid MPAL, and you see that there's a FLT3 alteration, I think that could be an early indicator that it could belong to this subgroup. But at the moment, we don't have, molecular diagnostic markers for it.
Great. Let's see, maybe going back to a question for you, Anthony. There's a question around HiChIP and kind of ancillary technologies there, like GeoPet and ChIP-seq. Can you just maybe compare and contrast the differences between the three?
Yeah, sure. So GeoPet, yeah, I had some experience with that in grad school, and that conceptually, that workflow is ChIP followed by Hi-C or proximity ligation. And in contrast, HiChIP is in situ Hi-C to capture those interactions within nuclei, followed by ChIP. In the paper that I published with Bing, we benchmarked the GeoPet and show some of the advantages of HiChIP in terms of the signal to noise of the long range interactions that you can detect. Using HiChIP, the fact that you need far fewer cells, I think we typically recommend a couple million cells for HiChIP, where I think GeoPet in the past has used far more than that. And then ChIP-seq, I think I had a slide in there about kind of what that is, and we'd like to envision that people would use HiChIP to obtain the ChIP-seq profiles and the long range interactions as a sort of a single, multi-omic assay.
Okay, great. I see we're just ticking past 1 minute past the hour, so I think that'll be all the time we have for questions. Just wanted to let everyone know if you had a question that didn't get answered, someone from Arima Genomics will follow up with you directly after this event. As we close the webinar today, just also wanted to mention that a survey will pop up on your web browser. We'd appreciate any feedback that you could provide on today's event, because we plan to host many webinars over the course of this year. I want to thank again our speakers, Anthony and Lindsay. Really appreciate your content, your presentation, and thank you all for attending today's webinar.